Pedro, for hosting this session, and thank you, Rebecca, Gianni, and Danuta, for offering me to give a lecture on food and NMR relaxometry. I just realized that although I have been teaching online for the last couple of months, this is my first webinar. Hope you will enjoy the lecture as we are going to talk about different food systems and how we can apply NMR relaxometry. Just a couple of words. Let me introduce myself for those who doesn't know. I am Mecit Oztop. I'm a faculty at Food Engineering Department at Middle East Technical University, Ankara, Turkey. And I've been in this cost action since the beginning, and I've been in the working group 3, which is environmental and food applications. And throughout that cost action, we had the chance to improve our NMR relaxometry knowledge significantly. And today I'm going to talk about at which point we are regarding how we can use NMR relaxometry, especially field cycling NMR relaxometry on food systems. Okay, so let us start. Well, let's first start with saying that foods are complex systems. So it's not really easy to develop an analytical method for a specific food. So you see here milk. It's a liquid, but it contains many different ingredients. It has proteins, lactose, oligosaccharides, vitamins, all of them having different functions. When you talk about eggs, this is a protein matrix where each protein has a different functionality. Cheese, it is a gel matrix where we have proteins trapping the water and other nutrients, and we have kind of a semi-liquid system, and it could go, we know that there's been very different forms of cheese just due to the processing time. Or you see over there a photo of butter. Butter is also a complex food system because the crystal structures in the butter determines the texture, and attaining that proper crystal structure is quite significant. Meat is another complex food system. Again, we talk about the protein matrix where we have water and other nutrients. Chocolate is another matrix where we have cocoa butter and cocoa mass, sugar, emulsifiers, just mixed in a matrix, and crystal structure is also quite important there. Or we can talk about a simple food, which is honey, but still honey contains many different components, different types of sugars. And when you look at the literature, you see that there are tons of studies about the quality and authenticity of honey. Or if you think about juices, fruit juices, although they contain significant amount of water, we still have a lot of other ingredients such as fibers, pectin, enzymes, which also create a complex matrix. So the take-home message is that we are dealing with a complex system, and coming up an analysis technique for that is quite difficult. But then the question comes, why do we analyze, why do we do the analysis in foods? Because we want to have good quality food. So we want to know what is going to affect the quality of the food. And we are caring about food safety. As food scientists, our major goal is to ensure to provide consumers good quality and safe food. So we are analyzing the food also in terms of safety. We are analyzing the food for authenticity purposes. We are trying to protect the origin of the food because we know that food fraud has been a significant issue. Adulteration is also another problem. To make foods cheaper, just different ingredients which should not be added in the original foods are being added, creating an adulterated food. So to address all these issues, we need to analyze the foods. And what do we use for analysis? There are a bunch of different wet chemical methods to do analysis. And there are methods that can fingerprint the foods, and the near-infrared spectroscopy, FTIR, Raman spectroscopy. So these are just some spectroscopic techniques which can help to fingerprint the food. Or there is this foodomics approach, and there is the chromatographic experiments, which can also quantify the constituents of the food materials. Well, but today we're not going to talk any of them. We are going to talk about the NMR. Well, and we are not going to talk about spectroscopy, because spectroscopy is also another technique that can be used to get fingerprints or to address the quality issues in the foods. And spectroscopic measurements are usually done at higher fields, but we can also perform spectroscopy at benchtop system with the newly developed instruments, and we can get quite good fingerprints. Over there, you see the spectrum, NMR spectrum of milk, which has been obtained at a benchtop spectrometer working at 1 Tesla. Okay, well, when we talk about food and NMR, so we have two aspects. It could be either the spectroscopy or we could do time domain NMR, we could do relaxometry, and we can do field cycling NMR. Okay, on the time domain NMR, what do we do? Usually when you look at the time domain NMR studies in the literature, you see that usually we are measuring the T2 relaxation times, spin-spin relaxation times. And foods, as I told you in the beginning, are complex system. So usually we have a multi-compartmental nature in the foods, which brings out the fact that whenever you have the T2 relaxation signal, and when you decompose it, you're going to obtain different proton pools, different relaxation time pools from the sample. And these relaxation times for individual proton pools could be attributed to the changes that occur in the foods. You see here the T2 spectrum of honey, where you see two components exist, and this is from a study where honey has been adulterated with corn syrup. And on the right, you see another spectrum that belongs to meat, where meat has been brined in different salt solution. And again, with these peaks, you see different proton pools. So this is not a signal that we analyze like in the spectroscopy. So these peaks do not identify individual chemicals, but here we talk about proton pools. Well, on the other hand, we can also do 2D NMR relaxation maps. So we can measure T1 and T2 at the same time. So we can obtain the information from the food, from the proton pools in a more detailed way. So summarizing all these, this classical TD-NMR approach provides us proton pools, which can be also associated with the cellular structures. What do I mean by the cellular structure? Plants, fruits, and vegetables has been studied significantly by food scientists, and we are applying different processes to these fruits and vegetables just to increase their quality. And whenever we are applying these processes, we are changing the cellular structure. And to assess the change in the cellular structure, these T2 relaxation times have been used a lot. So some compartments have been assigned to cell walls, some compartments have been assigned to cell membrane, or in the case of large plant cells, we usually associate, let's say, the longest relaxation time to the vacuole wall. So cellular structural information can be obtained. Fingerprinting by using a TD-NMR spectrum could also be an approach, or we can monitor the change occurred due to different ingredients, different processes. We can see the crystallization process, because we know that as crystallization occurs, relaxation times are going to change. So with the following the proton pools, we can monitor that. or we can check whether the food is adulterated or not. We can characterize emulsions, emulsifiers. Emulsions are very important food system. We have oil and water present in many food systems together. In order to assess the stability of these systems, we usually characterize these systems. TD-NMR or with the gradients, we use this approach even to measure the particle size of the emulsions, but also in general to have a general macroscopic idea about the stability of the emulsions. So these are the all things that we can do with TD-NMR. Today in my talk, I'm not just going to talk about TD-NMR. In fact, I will just have a couple of applications that we have recently done in our lab, but mostly I'm going to talk about the applications that we have done on field cycling NMR relaxometry. Well, this information is from the Stellar's application notes. Well, and in fact, I have met with FFC with this cost action. So, I mean, I've tried to recognize how we can use FFC on foods through Eurolex. And here you see the definition of the FFC for those who are not familiar. Well, the classical time domain NMR works at a permanent field, so we have just one frequency. On the other hand, FFC NMR is the only technique which permits the measurement of T1 spin lattice relaxation times over a wide range of magnetic field strength with just one instrument. Thus, it offers the complete investigation of the molecular dynamics in a variety of substances and materials. So we are measuring T1 at different fields because we know that T1 is field dependent, and we have the option to go to really low fields, and at these low fields, with the help of obtaining information at these low fields, we can assess information about the molecular dynamics. And for the time being, it might seem not clear how, in terms of food quality, we are going to relate this molecular dynamic information. We are going to explain it in the specific examples as we go on. Well, when you go Web of Science and just you click FFC and food, for the time being, you just see a couple of studies. This has increased by some number in 2019 to 20, especially after the Eurolax section, and we start to see some more studies, but still it is not that much compared to TD-NMR studies. So with FFC NMR, what can we do? We can have an idea about the molecular dynamics from very slow dynamics of the order of 10 of millisecond to the nanosecond range. We can have an idea about the time scale of the motion, and translational rotational diffusion is also another information that we can get. And how we obtain this? So the magnetic field strength is changed from 5 to, let's say, 5 kilohertz to 40 megahertz. Okay, how come this can be important in food? Okay, well, dynamics in food. Now I put here a slide which shows the water content of the foods. So you see the majority of the foods that we talk about has quite high water content. So water content and thus the water activity affects the properties of the food significantly. So the water content, the water activity is going to affect the quality, is affecting the shelf life, is affecting the texture, and as a result, all these effects due to water are directly reflected on the dynamics. So dynamics becomes important. So understanding the role of such an important ingredient in a food system will lead us information about quality, information about shelf life, texture, and also the ones that I listed in the beginning, authenticity or ensuring and even the safety. Okay, we are changing the food significantly. We are changing the foods by different processes. We are applying pasteurization to milk. We are drying the foods. We are pressurizing the foods to improve their shelf life, or we are obtaining glossy products like candies or to obtain desirable confectionery products. And in all these systems, the dynamics of the water is affected. Of course, here we should not only have the idea that with FFC we can just address the dynamics of water. Well, foods also contain significant amount of fat, oil, and there has been some studies which also show that, especially on the some oils, potential that FFC could also help us to understand, to differentiate, for instance, different oil types from between each other. Okay, so but then it comes to question, what is the main challenge of FFC NMR and food? Well, again, I'm repeating, foods are complex systems. And when we started to work with foods and FFC NMR with the physicists, usually physicists said that, okay, foods are complex system. Please describe us your system. Okay, as food scientists, we try to describe our system, but still it's complex. So that is why there is conceptual difficulties involved in the data analysis. Selection of the appropriate model is very important, and we are learning this. So, and we are trying to develop this database so that we will have the appropriate models for the proper foods. And another important thing is, when we are doing FFC NMR, we also need to have complementary physical and chemical measurements to confirm our finding. We are going to find a correlation time, but what will this correlation time tell us? Okay, it will tell us the dynamics, but will it be reflected on any other physical parameters? So that is why this is something that also has to be addressed. The complementary physical chemical measurements in that regard are important. Okay, so now let us see what has been done, what can be done with the NMR, with the FFC NMR methods so far. So now I'm going to specifically show you some examples from the literature and also some studies that we have done in our group with the help of the STSMs that have been performed in the cross-section. So unfortunately, we still do not have an FFC instrument, but luckily we have good collaborators, and with them we had the chance to get some data. Okay, so let's move on and start with the first example. So that is a study from 2011 from Pellegrino Conte. So this is a group that is publishing really good quality papers on FFC NMR applications on food, and this is kind of one of the ones that first came out. And here, different pistachio oils have been examined by using FFC NMR, and here you see the models that have been used, and translational contributions, rotational contributions have been addressed. So they are combined to fit the data, to fit the relaxation, the dispersion profile. And as a result, what has been found, some correlation times have been found, and these constants have been found. And for instance, AD also has information about the diffusion coefficient. And what is the outcome? The outcome is FFC NMR was able to differentiate the pistachio oils. So that is kind of one of the studies that has been, that I came up, and it has a really significant finding. And now I'm going to talk about another example. This is about a study which we are doing in our lab, and the FFC data for that study has been acquired in Professor Danuta's lab in Olsztyn. And there is some preliminary analysis, but we are going to improve the data analysis. But let me first describe you the concept. What type of food system we are dealing here? Well, here we have a protein hydrogel. A protein hydrogel is, especially from a food protein, is important because we can put some bioactives, we can encapsulate the antioxidants, vitamins, colorants, so that they are going to be kept longer. And we use here whey protein. Whey protein is an important protein in the milk. It is the soluble protein of the milk. The insoluble one is the casein, which usually leads to cheese. The rest, which is 20% of the milk protein, is whey. And by using this whey protein, we were able to come up with some gels. But these gels were not just simple gels. We put some extract in it. We put black carrot extract, because black carrot extract is an extract which is a good colorant. It has significant antioxidant value, and we want to keep this color of this black carrot extract, and we want to get use of the antioxidant activity. And while doing this, we did not just rely on the whey protein, but we also added polysaccharides to the gels. And we use 3 different polysaccharides. Again, these are food-grade polysaccharides: xanthan, pectin, and another Middle Eastern hydrocolloids, because this is mostly grown in the Middle Eastern region, especially in Iran and Middle Anatolia. We have this gum tragagans, and we created some composite gels. And here is how our gels look like. So we have a hydrocolloid, we have proteins, and we have the black carrot. So what is the function of this gel? We can use this gel to fortify the antioxidant content, for instance, of several beverages or soups. So we can have such an application area. But the important question, the research question that we ask is, you want to modulate how well the black carrot extract is going to stay in the gel, so how the releasability of the gel is affected. And in that regard, understanding the water dynamics becomes important, and we not only performed T2 CPMG experiments at 13 megahertz by using the system in our lab, but also conducted FFC experiments. So to characterize microscopic changes, to quantify the motional mechanisms in the systems. And the T2 relaxation times, in fact, the mono-exponential values did not show a significant difference. And we performed also some spectrum analysis by using the XPFIT software. But the thing is, the dispersion profiles showed us some information. So there were 3 dynamics. So we have three correlation times have been calculated. And here you see the models and the correlation times, and also the diffusivities. So here is kind of the summary slide. So correlation times of the black carrot containing hydrogels, and correlation time of the hydrogels in the absence of black carrot, because black carrot extract also changed the dynamics in the gel system. So depending on whether we have the black carrot extract or not, correlation times significantly affected. And we were able to obtain some information about the nature of these hydrocolloids, how these hydrocolloids are affecting the overall gel structure. For instance, in one of our previous observations, we said that when we have xanthan and xanthan added to the whey protein, and when we put the xanthan whey protein gel to the gastric environment, the gel just becomes so hard. So this kind of information now will be more elaborated by using the correlation times. And also diffusion coefficients have been calculated. So it was found that diffusion of the water was hindered in the gel matrices, and hydrogels have different diffusion coefficients. So now let's move on to another example, which is more like a clear food example. And in that case, I'm going to address honey. Well, in this one, in the first part, there is no TD-NMR. This is just another technique that we are going to use. But since the results really look promising, I want to show it. Well, here in the slide, you see two honey. One is liquid honey, and the other one is like crystalline honey. And normally in honey, we don't like to have crystals as long as we don't talk about cream honey. And what happens in honey is honey contains glucose and fructose, and glucose crystallize in honey if not stored appropriately, and then it's going to form granules, and that is what the crystalline honey is. And as you see over there, glucose solubility is lower than fructose, and that is the reason why honey crystallizes. And there are also some other factors like the amount of the water glucose ratio is kind of an indicator which can give you an idea about the honey crystallization. Well, in order to understand the crystallization, there are different approaches. Water activity could be followed, sugar profile could be followed, melting enthalpy by DSC could be followed, or NMR relaxation times could be used. And in this study, in addition, we use the magic sandwich echo sequence also to quantify the crystals. And we performed the experiments in two ways. In the first part, we just had the crystallization experiment, and we intentionally created crystals. So what we did, we added glucose powder to the honey, and then at every 2 hours, we followed the relaxation times, and also we measured this behavior through the DSC as the complementary method. Well, it's not only the crystallization that we did, we also followed the melting. So we put the already crystallized honey into the magnet, which is set at 55 Celsius, and then we try to follow the melting profile. Well, here you're going to see some plots, but I'm just going to summarize. First of all, NMR relaxation times show the presence of 2 proton pools. And we see that with enthalpy and with the relaxation times, we can observe some correlation. But there was a point where we realized that there's the formation of the stable crystals, and when we try to follow the crystal content by using this magic sandwich echo sequence and also the DSC result, we catched up this point. So after that a point, we had the stable crystals, so the content did not increase significantly, but MSC was successfully used to monitor the crystallization. And also you see on one of the plots on the top, T2 relaxation times also have very high positive correlations with the cooling time, with the crystallization time. Well, and now, by the way, we also have some FFC data on honey adulteration, but I'm not going to show that data because still it's under we're trying to assess the data. But I'm going to show you another study from the group in Poznan. So they are also doing very good food FFC studies, and where they looked at the crystalline fraction of the honey. And here you see the dispersion profiles. And what these dispersion profiles tells us is with increasing temperature, we observe the relaxation rate. We observe a decrease in the relaxation rate because we have liquefaction actually, and the viscosity of the honey is also decreasing. But there's a very important finding in this study, because once this honey is melted, or let's say heated to a high temperature, and later on cooled to the room temperature, the dispersion profiles changed. So the top 3 dispersion profiles that you see over there are the dispersion profiles that is obtained after the reheating. So what does this tell us? So if honey has undergone some heating, so that can easily be reflected on the dispersion profiles. But of course, in order to have this information, we need to have the clear idea about how the dispersion profiles will look like or which physical property of honey, but still it shows us a quite good potential on what happens to honey with processing. Honey is a product which can be subjected to adulteration or fraud quite easily. So catching this point by FFC NMR is really very crucial so that we can protect the authenticity, we can protect the origin of this honey by developing appropriate methods. Okay, now I'm going to move on to some other examples. We started with honey as a sweet product, and we are going to follow with some other confectionery gels, which are also sweet products. But before that, I just want to show you some information that we get, because this is going to be important while interpreting the confectionery gel data. Well, we did some studies on the hydration of the sugars, and just I want to point out that we are working with this rare sugar, the allulose. This is commonly worked in Japan. There is this rare sugar institute. They're producing it this high amount. This is a carbon-3 epimer of fructose, a natural sweetener, and lower calories since it is not digested. That is why now it is the time to look for how the foods will have a structure when we have rare sugar added. Because even though it is a carbon-3 epimer of fructose, its behavior with water is completely different. That is why the properties significantly change. And here in this study, what we did is we have glucose, fructose, allulose, sucrose. We prepared some 3 grams per milliliter samples, and we measured the relaxation times. And T1, T2 relaxation times were measured initially and also were measured after 1 day. And first of all, this hydration ability of the sugars has been studied by TD-NMR in a previous study published in 2012, and there the difference is attributed to the hydrogen bond length. Well, we don't know that information for the allulose, but what we know is that although allulose is highly soluble in water, the water binding ability of allulose is smaller than sucrose. So allulose, although a monosaccharide, it doesn't bind water it doesn't decrease the water activity as does sucrose. So this has been also shown in some studies, and that property could be desirable in certain formulations, could not be desirable in certain formulations, because whenever we add sugar, the function of the sugar is not just to make the product sweeter, but also it has a direct effect on the texture. Think about a low sugar cookie, for instance. This is not going to have a very desired texture, okay? You need to add something else to make it appropriate like a real cookie. So if the allulose is not doing what sucrose does, then we have to know how this can be prevented or what is the reason. So confectionery gels are high-calorie foods. How can we reduce it? Or how can we improve the nutritive properties? So we can add rare sugar, and that is what we have done. So we had gelatin, starch, pectin gels. So confectionery gels can be prepared by different gelling agents. Here you see we have gelatin, starch, and pectin as the gelling agent. And these are 3 different confectionery system. Gelling agent has a vital effect on the quality of the product because it affects the texture. Why people don't prefer gelatin? Due to the vegan preferences. Starch gels is really hard to make. It might be really hard if you do not process it properly. So in that case, then the texture is going to just address for some part of the consumer who like harder gels. So in overall, we have a gel system in a confectionery product, and if we want to prepare it with rare sugar, then the water dynamics is going to be different. So in that case, FFC will be a great tool to examine. So I'm going to show you 3 different FFC profiles. So for the gelatin candies, for the starch candies, and also for the pectin candies. For all these candies, we also have the physical measurements. We have the moisture content, hardness values, and texture profile values, color values, and in addition, we have the dispersion profiles. Well, in the first one, and the gelatin one, we increase the amount of the allulose, and as the allulose amount increased, the moisture content increased. And the NMRD profiles also reflected that. So we, as the allulose kept increasing, we had the lowest relaxation rate. The theoretical model was fit to a 2-proton pool model. We had the confined water, free water, and then the confined water was further described by the contributions of the translational and rotational motions, rotational diffusion, and the analysis was carried out accordingly. Well, what did the data analysis tell us? It was shown that increase in moisture content and decrease in hardness values with allulose in fact changed the translation and rotational dynamics. So they were all parallel with each other. Increase on the concentration of allulose led to an increase on the relative population of the pool of water molecules. So we really further moved one step further. We have seen that in the T2 relaxation results, but now we confirm that with the change in this correlation times. Starch-based confectionery gels were also examined. In this case, we do not have just a confectionery starch gel. We also have rare sugar plus soy protein isolate because we want to functionalize the sugar, and the dispersion profiles have been obtained. Still, the model analysis is going on that. These results have been acquired at Professor Danuta's lab. And what information we want to get here? We at least have some idea. We observed longer spin relaxation times with the presence of allulose, which was consistent with the gelatin results. And with the presence of soy protein, also the gel structure change. So all these dynamical changes will be directly reflected on the food quality. And pectin-based candies were also being studied. The FFC experiment in this case was performed at Professor Pedro's lab in Lisbon. And now still we are trying to analyze the system. We try to fit an appropriate model. Well, here you just see the T1 curves with respect to frequency. So we can identify 2 different regions, below 9 megahertz, above 9 megahertz. So the behavior was kind of changing. But still with the model, we were able to see that the presence of allulose, presence of rare sugar is significantly changing the dynamics. And our normal physical quality measurements show that if you want to make a pectin gel with the allulose, it's not going to be a good gel. Okay, authentic Turkish delight. Well, this is another question that we want to ask. Turkish delight is very important for us, for Turkish people. So we want to protect its authenticity. Turkish Food Codex, the regulations say that we only should have sucrose in the Turkish delight. But this is usually not the case. To make it cheaper, usually corn syrup could be added. Addition of corn syrup might improve the physical properties, might prevent crystallization. You might obtain better, you might obtain starch at a much shorter starch gel, the Turkish delight at a shorter time, but still it is not appropriate for the authentic Turkish delight. So in that regard, we prepared Turkish delights having different corn syrup content. And in this time, my student went to Poznan at the Ratokis lab to do some FFC experiments. And here you see the formulations of these starch gel, but we use also different corn syrup, which is available in the industry, and we obtained the dispersion profiles. Well, the dispersion profiles were obtained at 2 different temperatures. One was 25 Celsius the room temperature, and the other one was 4 Celsius. Although Turkish Delight is not stored at 4 Celsius, we wanted to select this temperature because Turkish Delight contains starch, and starch retrogrades. What does retrogradation mean? It is like recrystallization of the starch. Bread stales due to retrogradation. This is the same thing. Also, we have the Turkish Delight, which could have starch recrystallization, and that is why we were able to, we want to see this change. Again, the data analysis is still going on, but what is going to be the research question, what is going to be the final question we want to answer is, will we be able to say that, okay, if you have a Turkish Delight, well, there is this characteristic that you have corn syrup. If you're able to obtain that, then it's going to be, of course, a quite good outcome. It's not easy. We really need to perform the experiments on a good data set, but still we see, at least from the results, that there is potential on identifying the presence of the corn syrup in the Turkish Delight. Well, we also have previously a study, again from Professor Pedro's group, to differentiate Frankfurters of chicken, turkey, beef, and pork origin, and the NMRD profiles were obtained. Well, the results were not exactly 100% conclusive, but at least we had the potential. This study was done on the ready samples, so this has to be repeated on a controlled sausage, where we will have you will know the matrix in advance, and then we will be able to obtain much better information. And 2 more other studies that I have recently come up. This is from Philip Woodard, where the manganese ions in wine were determined by NMR Relaxometry. So the results really were able to show that the technique has this potential. And finally, there is again another recent publication from the contest group where they use the FFC NMR during drying of the apples. And in fact, 2 types of drying have been followed: microwave and hot air drying. And the conclusion that they were obtained was, for instance, in microwave drying, the apples were, let's say, shrinking differently than hot air drying. So what could be the reason? They use these approach, simple, the free model approach, to calculate the correlation times and fit the data, and the behavior shown by FFC, where they said that could explain the lower shrinkage in apples by NMR, by microwave drying, compared to the convective and hybrid heating. Okay, so let me make my concluding remarks. FFC has a potential in foods, but what we are struggling more is food scientists and physicists should work together for the data analysis and interpretation, because for physicists to imagine the food is difficult, so we definitely need to work together. And especially if we want to expand FFC on the instruments, make it more wide, then this software that we are going to get the data should also guide the food scientists for the analysis, so that they will be able to get some quick interpretations. Okay, I just would like to acknowledge all the contributors who helped during these SDSMs to my students, and also to my lab members who have been involved during these projects. So I would like to thank you, and I will be pleased to get your questions. I think I'm on time, Pedro, right?